The human brain is the most complex organ in the human body. The brain controls human behavior, body movement, and is the seat of intelligence. It is made up of billions of neurons which transmit information from different parts of the brain using electrical impulses. Brain activity can be monitored by placing electrodes on the scalp to measure the small electrical potential, and this is known as the electroencephalogram, or EEG. EEG has been used by clinicians to diagnose diseases such as stroke, epilepsy, and depression. In the bioelectronics field, a non-invasive brain stimulation technique has been widely used to treat these diseases. The technique is called transcranial alternating current stimulation, or TACS. It is done by passing through current in sine wave via electrodes onto the scalp. The TACS injection will change the action of the neurons in the brain. Recently, the effects of TACS injection onto the underlying EEG can only be analyzed before and after stimulation. Direct analysis during stimulation is not possible. The EEG cannot be observed because it is completely obscured by large stimulation artifacts. As shown in the figure, EEG is visible from 0 to 30 seconds. TACS is injected at the 30-second mark until the 90-second mark. It is obvious that the TACS artifact completely obscures the EEG, making it impossible to monitor. Our aim is to remove the TACS artifacts using robust and efficient methods. There are two approaches to achieve this, by investigating both the software-based system and the hardware-based system. The first objective is to create a computational head model. Secondly is to design a hardware simulation for EEG collection. Finally, to create robust algorithms for artifact removal. For the computational head model, we have built a 3D model using finite element method. This is done using an open source tool called ROAST. After creating the FEM model, TACS electrodes are placed onto the scalp. TACS is then injected onto the model. After the simulation is complete, ROAST produces a result file that contains the voltage due to TACS data at each voxel coordinate of the FEM model. The TACS voltage is then extracted from this file, scaled and added to the EEG signal. After several steps of signal modulation, an EEG and TACS signal is produced. To measure the EEG and TACS signal efficiently, we have designed a novel type of electrode called tripolar electrode. It consists of 3 rings which are divided equally to ensure better spatial resolution. This novel electrode is better than standard ones because it has higher accuracy and better localization. To further process the signal and remove noise outside of the bandwidth, an EEG electrode circuit design has also been proposed. For the artifact removal algorithm, we have created a novel method which combines a signal processing algorithm called singular spectrum analysis with a machine learning technique called spot vector machines. Let us assume that the EEG and TACS signal is signal A. This is the input for our SSA and SVM method. Firstly, signal A is decomposed by the SSA algorithm. This will produce the reconstructed components of signal A. The machine learning SVM model will classify which of the components are artifactual and which are not. The components will be labeled accordingly, separating them into 2 different classes. The artifactual components will be eliminated. The non-artifactual ones are reconstructed to produce a clean EEG signal, which is free from artifacts. In the end, the whole process will produce a clean EEG output signal without any large stimulation artifacts. Our project has succeeded in creating methods to remove TACS artifacts from EEG recordings by implementing novel methods of artifacts removal. For future development, our hardware design could be fabricated and physically tested. Removal algorithms could also be enhanced to remove different types of artifacts. Finally, the current computational head model could be developed into a multi-scale model.